Good morning, everyone. Welcome to this webinar where we'll discuss use of Pbpk modeling in pediatric drug development. Many of the concepts and strategies that we'll talk about are applicable to pediatric modeling in general, but the technical aspects and the examples that I will be presenting are going to be specific to the GastroPlus software. So in this presentation, I'm going to discuss or try to address three basic questions. Why we do Pbpk modeling? How we do it? We will talk about some of the physiological aspects and how to scale different processes from adults to children. And finally, what? We will address some of the examples, some of the case studies, and including case studies that were used in the regulatory applications. So why Pvpk model? We do know that pediatric drug development is challenging. There are ethical considerations for including children in trials. There are difficulties recruiting the patients, getting parental consent. It is a heterogeneous group from neonates all the way up to adolescents, and different considerations may apply for each of them. So the development is to a certain degree always going to be dependent on extrapolation. And this is where Pbpk models are really powerful. So what is Pbpk model? It is a set of mechanistic equations that combine the properties of the drug, the properties of the formulation, and the properties of the system, in this case, properties of the physiology, to simulate and predict how that specific compound within that specific formulation is going to behave when placed within that given system. So we can use the data in adults to parameterize these relationships, to parameterize and verify that we are correctly capturing all of the relevant mechanisms. For example, capturing the interaction of your compound with specific enzymes and transporters, or how the compound will be dissolving and getting absorbed under different conditions by properly parameterizing the ionization and solubility aspects and so on. And if the model is parameterized correctly and the processes are captured mechanistically, then we can simply replace this part of the model with a new physiology. We can, instead of the adult physiology, we can place in parameters representing a physiology of a specific child and start predicting how that compound and formulation would behave when administered to a child instead of the adult. And we know that this is not just an academic exercise. It does have real practical applications that have been recognized by regulatory agencies around the world. Here are excerpts from several guidances. For example, this one is from US FDA all the way back from 2014, where they already recognized that modeling and simulation should be a part of all pediatric development programs, and highlighted specifically some of the applications of Pbpk models. Right? First, the dosing recommendations for that first in pediatric study, optimizing the study design, recommending the doses, and so on. In 2018, EMA published their guidance on general physiologically based pharmacokinetic modeling. And pediatric dose selection in pediatric trials or for children has been highlighted and as one of the common or one of the main purposes of the Pbpk models. And specifically, the prediction of the PK in pediatric patients is one of the high regulatory impact analysis where it could have labeling consequences. In 2020, Japanese PMDA released their guidance on Pvpk modeling in general, and again, predicting or setting the dosage recommendations for clinical trials in children is one of the most common or one of the critical aspects of using the Pvpk modeling. And finally, last but not least, hot off the press, just about 2 weeks ago, CFDA released their guidance, which is now specific for use of Pbpk modeling in the pediatric applications. So again, different markets, different agencies from around the world, they are all recognizing the use of, or the utility or the value of Pbpk modeling in the pediatric drug development. And they are not only willing to consider the applications, they are actually expecting it. So of course, in order to do that proper prediction or to be able to do all of that, we really need to know this piece, the system changes, how the physiology is changing from neonates all the way to the adults. And that is what allows us to then predict how the compound will behave or how the compound and formulation will behave in different physiologies in children of different ages. So for the next few slides, we are going to talk about some of the physiological considerations. And more specifically, we will talk about the physiological considerations or aspects that we have incorporated within the GastroPlus applications or within the GastroPlus software. I'm assuming that most of you have seen a diagram showing a physiologically based model. It is a collection of number of compartments which represent individual tissues in the body, and they are connected by, in our diagram, we have the blue arrows showing the blood flow. So to make this a really physiological model, give those compartments physiological meaning, each of these little boxes, each of these compartments represents a specific tissue in the body. And so it is characterized by a specific volume for the tissue, by a blood flow, that specific perfusion rate for the tissue, composition of that tissue, how much water, how much lipids, how much protein does it contain, and of course the enzyme and transport expression levels in those tissues, and other characteristics as relevant for the disposition of the compound. One of these boxes I'd like to highlight a little bit more, and this is this one representing the gut compartment. Very important, especially for orally administered compounds. Gut is not a homogeneous single compartment. The environment is very different between stomach, different regions of the intestine, all the way down to colon. So if we want to properly account or predict how an orally administered compound or oral formulation will behave in children of different ages, it's going to be very difficult if we were relying on a simple Ka value. If we really want to do that scaling from adults to children, we do also need to account for changes in that intestinal physiology. How quickly the compound will transit through the intestine, how much time is it going to have to dissolve in child versus an adult, what kind of environment is it experiencing, how much volume does it have available to dissolve in, what is the pH, what are the concentrations of bile salts, and again, what are the expression levels of enzymes and transporters that will be affecting the behavior of the compound. So all of these, again, for the intestine specifically, need to be accounted for the same way as for all of the other tissues in the body. I'm going to show only few pieces of information just to give you an idea about what kind of information we've used to actually characterize or parameterize those physiologies within GastroPlus. And these are, as you will see, these are all information from literature, from various public sources. So other models would be using very similar information to parameterize the models, but the actual equations might, of course, vary. So this is showing examples of parameterizing or changes in body weight and body height versus age. And these are shown specifically for American population. We do have also separate set of data and characterization for Japanese and Chinese populations. It's just so happened that in these slides, I do have the plots created for American population. But again, it would be similar plots showing how the body changes in terms of height and weight. When we are trying to predict the PK in children, we are not going to be looking only at a single child usually, but eventually we will want to predict a PK in a group of children, doing a population prediction. So the algorithms need to account not only for how the average child would be growing, actually I should be looking mainly at the curves up to about 20 years, how the average child would be growing, but also be able to account for the variability in body heights, body weights, as well as the different tissue sizes. And this slide is showing just an example of populations created by our population simulator, so virtual populations of subjects. The first column is showing adults, 20 years and older. The middle column is showing children to adolescents, 1 through 20 years. The right column is showing infants, 0 to 1 year. The top row is for female, bottom row for males. And in each of those plots, you can clearly see two sets of data with slightly distinguished colors. What those mean, one of those sets is the actual data coming from the enhanced database, and the other set is showing the data that our virtual populations, our population simulator created, what kind of virtual population it will generate using the algorithms. And you can see that it is really able to very nicely capture the population variability. So it is able to generate a very realistic population of virtual patients. But going back to the individual tissue sizes and compositions and so on, of course, that is what we need to capture in order to predict PK. Right? Body height and weight are important, but where the compound is distributing is into individual tissues. Right? So we need to have correct algorithms not only for the overall height, weight, and BMI of the child, but also the sizes of the individual tissues. And I'm not going to go through every single one of those tissues. This is going to show only few tissue examples. Here I'm showing the body fat and heart weight as it's changing from children all the way to adults. This spike in here, that's about 1 year old. As babies, when they are born, they have less fat composition. During the first year, as they are growing, they are adding more fat. At around 1 year, they start moving and start losing some of that fat again. So that spike is the real and physiologically realistic. So we have similar plots and similar pieces of information collected from various literature sources to be able to parameterize the changes in individual tissue sizes for the physiologies. And again, in these figures, they are simplified a bit because they are shown only as only as a function of age. But of course, then going back to these plots, it's not going to be just the age, but also body height, body weight, potentially body surface area or BMI, right? Different tissues correlate with different overall physiological parameters. So all of those are taken into consideration. The blood flows, again, similar pieces of information collected from literature, specific for males and females, as well as specific for different ages. This is showing just samples, one of those data sets. I'd like to do a little word about the infant physiologies, where, of course, things get a little bit more complicated. Not for all of the aspects, though. When we were looking at creating the infant physiologies for children up to 1 year old, for many of the aspects like total body weight, height, or many of the tissues, it is a very straightforward relationship where, for example, here showing the liver and kidney weight. The green line is showing the 40 weeks of gestation. So these are shown as a function of postmenstrual age, so accounting for gestational and postnatal age. And you can see that for many of these, it's a smooth transition going from fetus into child, the body or the tissue just continues growing. But it's not the case for every tissue and every physiological parameter. This is showing, for example, the adiposity or the percent fat mass, where both the gestational age as well as the postnatal age, the age after birth, are playing their own separate roles. For on the left, the plot is shown for babies born at full term, and the blue is showing the data for boys, the red for girls. And the red and blue curves are corresponding to the black, solid, and dashed lines on the plot on the right, where the data themselves, the points, actually represent preterm born babies. So you might be thinking, why are they to the left? It is because here that gestational age actually does influence how that fat mass is increasing. And in the babies born preterm, it's actually increasing a little bit faster than in the term-born children, up to about the 3 months where they come together and then continue developing about the same rate. In addition to tissue sizes, of course, we are concerned about changes in the tissue compositions. Depending on how much water, how much lipid, how much protein is in different tissues, that will influence the affinity of compounds of different properties to that tissue. Compounds that are more lipophilic will preferentially partition into the adipose tissue. Right? The compounds that are more hydrophilic would be going more into the tissues that are more water, less lipophilic. Right? So again, having the composition is also important for correctly predicting the tissue plasma partition coefficients. And it's not very dense or a very rich set of data, but we were able to find some information in the literature in how roughly the tissue compositions are changing over age. And here we are showing, and actually, again, we are seeing most of the changes within that first year. These are showing from 24 weeks postmenstrual age. So these would be about 16-week premature born babies, all the way up to first year. Actually, no, these ones are going up to 18 years. And you can see that, especially for adipose, the changes within that first year, which would be about here, are actually quite dramatic. More dramatic, of course, for the premature babies, less for the term-born babies, right? But especially the very premature babies, even adipose tissue is mainly water, shown by the blue curve. And over time, it is changing more into the composition of the lipid, shown by the purple curve. Right? Less dramatic for the brain tissue or some of the other tissues. Again, we are not going to go through every one of these tissues. This is just an example of that type of information. Down here, the plot is showing how the total body water is changing. So again, similar information, how it's changing over age. And again, most of the changes or the most dramatic changes happening within the first year. Blood composition. This is something that you might be aware of already, especially within the first year. In this case, the plots are showing from the PMA of 24 weeks to 94, so that would be about 16 weeks premature newborn up to 1 year old, and showing significant differences or changes in both the AAG as well as the albumin in the plasma, and also significant changes in the hematocrit. Just to give you an idea what you are looking at here at this plot, the red is showing how the hematocrit would be changing in the fetus, and it is steadily increasing. The blue lines are showing what is happening to the hematocrit shortly after birth. So this light blue line that was showing about 26 weeks so very premature baby after about, actually it might have been about 30 weeks. This one maybe about 34 weeks birth. This one would be, the dark blue line is showing babies born at term at 40 weeks. And you can see that at every age, shortly after birth, the first few weeks, there is a significant drop in hematocrit. And then it starts again increasing until it reaches the adult levels. And so these parameters, of course, the changes in the plasma protein levels, changes in the hematocrit, are going to affect the fraction unbound in plasma and the blood to plasma concentration ratio. So to make things simpler, of course, we incorporated these relationships, and in addition to that, we've also incorporated scaling of FUP and RBP to account for these changes in the protein levels and hematocrit. This is just to make it simpler for you. We assume that more likely you would have the blood to plasma concentration ratio and FUP measured in adult blood. But if we know how the protein levels are changing, how the hematocrit is changing, we can scale it from the adult blood down to the pediatric blood, down to the age that you are trying to simulate. And the program does show you all the details, how that scaling was done. This is showing validation for the fraction unbound, since there were some data available for babies or infants 1 day to 4 months old, showing these are the predicted versus observed. For this scaling, we are using equation that was previously published. And for those of you who might be familiar with that publication, you might know that the authors looked specifically at the major binding protein, whether the compound is binding mainly to albumin or alpha acid glycoprotein. In our equation, we have lumped them together as a total protein. And, even though they do have very different absolute levels, but if you look at the levels as a percent of adult levels, so scaled, the ontogenies are actually quite similar between those two proteins. So for scaling down to pediatrics, we actually got very similar correlation as the authors did in this publication, where they accounted for the major binding protein. So a lot of information that needs to be compiled and put together to calibrate or to parameterize the physiologies themselves. And we haven't even talked about the enzymes or transporters. Right? This was really only about the body composition, tissue sizes, overall body size. But all of that information you do not need to generate or collect it yourself, because of course the algorithms in the Pvpk modeling software like GastroPlus asks you only for the specification of what physiology you want to do. Do you want to create physiology in human? For populations, you want to sample from American, Japanese, or Chinese population, male or female physiology. Right? We are also including different health conditions. Right? Since we are talking about pediatrics, you'd be more interested in looking at the children of different ages. Right? And again, you can go down to create physiologies for newborns born premature up to 16 weeks. Right? So all the way from 16-week premature newborns up to adults, you can create those physiologies. And all you need to define is what conditions you want. You can also define what body weight or BMI you want for that child. And the program will calculate the entire physiology. All the algorithms are already in the program. And the program would tell you what would be the body tissue sizes, compositions, blood flows, and so on for that specific child. As a side note here, there is a capability actually in GastroPlus through our mixed multiple doses profile. For those of you who have some familiarity with GastroPlus, you might know that we have this mixed multiple dose functionality that allow you to have irregular dosing, changes the doses over time, change between fasted fast physiologies and so on. But it also allows you to account for the growth of a child. And this would be more critical for looking at the children that are, or looking at the infants or neonates, where even from one day to the next, the physiology can differ quite a bit. So it does allow you to sort of schedule or to create that growth and create the growth exactly as you want it, because you'd be telling the program how the child is growing from that one day to the next. So all of this was heavily focused on the tissues within the Pvpk physiology. We haven't talked yet much about the intestinal physiology. And there is more limited information. Some parameters we are able to characterize and scale pretty well. Stomach pH, significantly higher in newborns. Then within a few days, it goes down in the fasted state. The length and radius of the intestine, subsequently volume. The enzyme transporter expression levels, again, ontogenies, are evolving. So those that are already known, like 3A4 and a few others, are incorporated in the program. Others we keep incorporating gradually as we go. But there is also still some information that is missing. Some of those are the pulse salt compositions, or how the permeability might be changing, structure of the villi, which affect the absorptive surface area, and so on. But again, it's still evolving areas, and as the new physiological information is coming up, we keep updating the physiologies to keep making the predictions better and better for the future cases and indications. So putting that all together, and again, a lot of the information that I was showing, especially the experimental data that we were collecting from literature, right? That's public information. This is just summarizing how we put it together within GastroPlus, what kind of physiologies you have an option to create. So we do, within the same framework, within the same program, of course, you can create the physiologies for animals as well as human, which makes it very easy and very quick if you have to go all the way from first in human prediction. You don't have to switch the program, everything is in the same one. And then as you are going from adults and want to predict the children, again, you just need to change the age and create a physiology within the same program. So when it comes to human, we do have the age-dependent physiology. You can define specific age, gender for the subject, health status, health conditions. And we also have those populations parameterized, or the physiologies parameterized for American, Japanese, and Chinese physiologies or populations. So that was a sort of a top level overview on the physiologies. And now we are going to spend a few minutes talking about specific mechanisms. How we would be scaling specific mechanisms responsible for clearance or distribution of the compound. We'll start with metabolic clearance, and quite well established. The CYP enzyme ontogenies, we are using the in vitro CYP enzyme ontogenies as published back in 2006. These are showing for most of the enzymes, for most of the CYP enzymes. I think there is one or two missing. And of course, also including that well-known switch for the 3A enzymes, where in the neonates, it is mainly the 3A7 form that is responsible for the metabolism of the compounds. And within about a year, the CYP3A4 becomes the dominant, and CYP3A7 is the less prevalent enzyme. Of course, it's not only about the liver, it's also the intestinal enzymes. And here, the additional aspect is that the enzymes in the intestine not only have different expression than in liver, but may also mature at different rate. And based on the information that we've seen, this is actually one of the papers. And I believe in here in the table, there was also intestinal CYP3A. They do, the intestinal 3A4 enzyme or 3 enzymes seem to mature at a bit slower rate than the liver enzymes. And here is one of the classic examples just showing how that ontogeny is working, where the model for midazolam initially calibrated from the in vitro and verified against the adult in vivo data was then used to predict PK in children. This was a virtual trial which included children 1 through, I believe, 12 years old. Right? And when I say it's quite classic example, you can see this has been presented and has been available since about 2008. So quite a long time. But of course, we didn't stop there. In 2015, we did have collaboration with University of Florida to see if we can calibrate or if we can verify and validate the ontogenies for some of the other enzymes. Right? And again, the focus was on the CYP enzymes. And the strategy here was taking the clearances from the literature, collecting the clearances for specific substrates of different CYP enzymes, caffeine and theophylline for 1A2, of course, midazolam for 3A4, warfarin for 2C9, omeprazole for 2C19, and so on. And first, building adult models, and then calibrating and validating the adult models, and then using those models to predict the PK and predict the clearance in children of different ages. And here are the results. And considering the variability in some of these enzymes, right, you can see, especially for 1A2, a lot of variability. More data was available for theophylline. Right? Prediction is actually pretty good, right? We seem to have a little bit of under prediction of clearance around here between the 5 and 10 years, right, within this range, which might be related to the fact that there actually wasn't much data available here for that age range. So it is an approximated or interpolated profile, but we don't really have much information about how that ontogeny is changing there. It still does give a very, very reasonable prediction. The other enzymes seem to be matching pretty well. Here are the cases for 3A4, 2C9, 2C19, 2C18, 2C19, which also had the more rich data sets. And those also had the ontogenies captured a little bit more completely, more accurately. So it resulted in more accurate clearance predictions. And once all of these enzymes were calibrated or validated, verified that the ontogenies are actually predictive of the in vivo clearance, we then used that model or those ontogenies to predict the clearance for acetaminophen, which is metabolized by several different enzymes. And you can see here the model was again initially calibrated and validated against the adults. And then the same model was used to predict the PK in adolescents, in the infants, as well as in the neonates by simply replacing a physiology. And you can see they did a very nice job going across the entire age spectrum, even for a compound which is metabolized by multiple enzymes. And this one it's not only about CYP enzymes. This was actually one of the early examples of pediatric Pbpk prediction. It was more than 10 years ago now, and it was modeled for oseltamivir, for Tamiflu, where they had to create actually quite complex model because they needed to model both the parent compound, shown in these lighter gray points, as well as the metabolite, oseltamivir carboxylate, and that metabolism is happening by the esterase. But through the strategy or through that learn and confirm paradigm, building adult model first, verifying strategy also in the animals, building animal and juvenile model, that gave them enough confidence that they know how to predict or how to scale even these clearances and this PK into children. And this is showing data for 5-year-old. Again, the top is showing the oseltamivir itself, and the bottom is showing the oseltamivir carboxylate. And the information still keeps coming up for the new enzymes. This is one of the more recent publications showing the ontogeny of UGTs, of covering most of the major UGTs, going all the way from, again, infants up to adults. And it came only about 2 or 3 years ago. So very good information coming up that is being incorporated in the program as well. If you look at the different elimination pathway, renal secretion, and specifically passive renal secretion, where the secretion is limited or dependent on glomerular filtration. So this is validating our functions for glomerular filtration rate and how that is changing with age, so the ontogeny for the GFR. And this is a quite well-known accepted change in the GFR with age, especially going from for the older children. We'll be talking a little bit more about the infants. The situation is not quite as simple as this picture would have you think, but we'll discuss that on the next slide. But for the older children, after a few weeks, it is fairly straightforward. And this is showing validation. And again, example that we've had or that we've been able to validate a long time ago, again, showing the prediction for gabapentin, which is predicted or which is eliminated by renal secretion, simple passive secretion. Model was initially built and validated in adults, and then used to predict the PK in children. The average child was about 7 years old. So let's take a look at what is happening here within those first few weeks or few days and weeks after birth. As shown on this plot on the left, the situation is not as easy, not as straightforward. What we are looking at here is the GFR versus post-menstrual age. So again, lumping both gestational age and the postnatal age. And the different colors are showing neonates born at different times. If we go from right to left, the red curve is showing term-born children at 40 weeks, the orange one at 38 weeks, the green one is showing at 36 weeks, 35 weeks, and so on. And if you followed the individual points, not just the lines, you would see that for each of those colors, shortly after birth, the GFR goes up. So clearly, the birth is what causes that increase in the GFR. But it is not necessarily the case for every premature newborn. It is the case only for premature newborns born at least 33, 34 weeks of gestation. For the younger ones that were born after just about 27 weeks, the GFR is first increasing at a very slow rate before it takes off. That is the time it needs to complete the nephrogenesis, so there is that additional complication. And again, we have validated these equations that account for both the gestational age as well as the age post-birth. And this validation is shown with a vancomycin compound, which again is eliminated by renal secretion. And glomerular filtration is the good indicator of its clearance. The plots down here are showing PK, the dark blue points, after the first dose, as well as steady state of vancomycin IV administration in premature infants. And the light blue line and points are actually showing the amount secreted or excreted in urine. And you can see that it's predicting well not only the PK, but the actual clearance very nicely. In the plot here, you would see the clearances, the renal clearances. We did not have full data, but we did find a publication reporting the clearances from population PK model. Those are the empty diamonds or open diamonds, the blue points here. And the red points are showing predictions for few of the subjects of specific physiologies from our algorithms in GastroPlus. And we did not have the exact information about the gestational age versus postnatal age. All we knew was what was the total post-conception or postmenstrual age. So we've done a couple of different calculations. The blue one here is showing a 2 days old babies, so basically newborns, at a different gestational age. And the purple line is showing babies born at 30 weeks and different postnatal age. So this would be a 5-week-old that was born at 30 weeks. This would be a 10-week-old that was born at 30 weeks, and so on. And you can see that those two borderline cases very nicely capture the overall range of the clearances from that population PK analysis. So again, helped us to validate that those predicted GFRs are actually accurate and correct. Finally, the last mechanism, let's take a look at some of the transporter effects. And when it comes to the transporters, of course, we are dealing with the aspects of distribution into the tissues, uptake and efflux transporters, as well as potential clearances. If, let's say, we are talking about the liver, there could be also secretion of the compound on the apical side into the kidney, into the bile, or potentially into the kidneys or into the urine if we are talking about the kidneys. And there is a lot of development or a lot of research currently done on characterizing the transporter ontogenies in different tissues by several different groups. Just as an example of how this field is evolving, this is a slide from one of all the presentations that I've prepared about 10 years ago. It was 2013. I believe it was for one of the AAPS workshops, where I was tasked to talk about the pediatric predictions for a transporter-related workshop. And looking for ontogenies, there wasn't really a whole lot. There was some information for PGP and maybe a couple of additional transporters, typically mRNA. A lot of qualitative information, just saying that, it's lower expression and so on. And there was information on the clearances for different compounds that could be used to infer what the ontogeny might look like. For example, Paraaminohypurate, which is renally secreted compound, and it is both GFR as well as the active tubular secretion. Could be an indication or changes in clearance of that compound could potentially be used to infer how certain transporters might be changing, to infer the ontogeny of the transporters, and maybe use these changes to scale the clearances for other similar compounds. Looking at this today, and as I mentioned, there is a number of research groups that are measuring transporter expression levels in both adults as well as in children. And this one is showing journey of just one of those groups. And where in 2014, so this was just about a year after I was trying to put this presentation and wasn't able to find much, where they had a publication on showing the hepatic and intestinal ontogenies based on the mRNAs. 2016, two of the publications from the same groups, one still focusing on an mRNA, but the other one now looking at the protein expressions. And this was focused on the hepatic transporters. 2018, 2 years later, again, hepatic transporters with even more focus on the very early days, fetus and newborns. Right? So really trying to characterize that complete age. 2021, one of the latest publications from them showing now moving into intestinal transporters. Actually, in this paper, they looked at both intestinal transporters as well as metabolizing enzymes. And in this case, looking at the protein levels. So, and of course, this publication does now have quite a few more transporters and enzymes than the early publication would. So it is evolving a lot of new information and keeps coming up and needs to be incorporated in the physiologies gradually as it's coming up. That being said, we can take a look at some of the examples, and I should say this information not only needs to be incorporated in the Pvpk physiologies, but we do gradually incorporate the ontogenies as they are coming up in the GastroPlus software. But some of these examples were actually done even before those ontogenies were available and can provide some insights into the importance of some of these transporters, or could have provided some early look at what the ontogenies might look like. The first compound here is valsartan, which is eliminated by biliary secretion. So critical transporters, the OATP 1B1, 1B3 for uptake into the liver, MRP2 efflux into the bile, and MRP2 also influences the absorption. It is the efflux in the intestine, and it has some impact on the kidney. But again, because the OATP 1B1, 1B3 is bringing drug into the liver, the hepatic or the biliary secretion is the major elimination pathway. And for this compound, we've actually done the full learn and confirm route of predictions, where we had data in vitro, had the PK data available in rat, in human, in adults, and then used that model to start predicting the PK in children between 1 and 16 years old. So the first step was also trying to look at the predictions based on the in vitro and animal data. So the first step we did here was take the in vitro measures of the transporter KMV max values, predict the PK in rat. Worked beautifully. So we knew we can go ahead and do prediction for human. Wasn't as perfect, but still very good first in human prediction. So once we had the IV, and we've seen that the IV was predicted very reasonably from the in vitro data, the next step was predict the PO, or oral administration. This is where some of the additional mispredictions started occurring. Okay? So before we would use this model to predict the PK in children, of course, we would want to refine this. Right? After every step, when you get the prediction, after every step of the prediction, once you get the observed data or the clinical data, you want to incorporate it in the model first to have the model as good as it can possibly be before you move to that next prediction step. Okay? So at this stage, we refined the model. We updated or refitted the passive diffusion through the tissue membranes and intestinal permeability to get the model that much better captures the shape for both IV as well as oral administration. And now we had a model that we would have more confidence to go into pediatric populations. And of course, since this is all retrospective analysis, we did have the data and we had the luxury of going from the older children into the younger ones. So we've been looking at this, starting with predictions for the oldest children, and this was 12 to 16 years old group. And if there was any misprediction, we could do the model update first before we would go into the younger group. But for here, the 12 to 16 years old, they were predicted very nicely. So clearly the expression levels of the transporters by this age appear to be close to the adults. So we were able to move to the next group, 6 to 12 years old, still predicted very nicely. So again, appears that by this age, by those 6 years, maybe the transporter again, already very close to adult values. 4 to 6 years old, still very nicely predicted. Finally, we got some apparent misprediction around the Cmax here for the youngest group of children, 1 through 4 years old. Now, of course, we could debate as to whether there really is misprediction here or not, because it's sparse data in children, and especially as we are going into these young children, the sampling really isn't dense enough to get even close to the Cmax value. So we could dispute whether this really is misprediction or not, but we do have additional information that, even though qualitative information, that the villi in the intestine are underdeveloped in children up to about 1 to 3 years old. So in this case, we actually didn't do anything with the transporters, but we adjusted the absorptive surface area. If the villi are underdeveloped, the absorptive surface area in the intestine would be smaller than we would expect for those very young children, about 1 year old. And that could actually result in slower absorption and would bring that prediction right in the realm of the observed data. So that might have been an indication or explanation for that misprediction in here. For the next validation example with these mechanisms, or for the transporter-related compounds, we can take a look at the amoxicillin. This compound has different clearance pathway. It is eliminated by renal secretion. And renal secretion means glomerular filtration and active secretion. So transporters involved in the renal secretion, uptake into the kidneys and secretion into the urine. But there are also transporters involved in other tissues. For example, oral absorption is also dependent on Pepti-1 and 2 and transporter, and then MRP4 moving into the blood and so on. In this case, we did not have animal data. So we were using just the human data, used human in vivo data to calibrate the model, and then use that model to predict the PK in infants. We did have quite a bit and quite rich data for this compound, including several different IV doses and corresponding amount of the compound secreted in urine. So had good rich data set to calibrate with confidence both the systemic distribution as well as the renal elimination. Had the oral data for several different dose levels that again allowed us to calibrate the saturation of that intestinal uptake transporter. And that is shown here, the less than dose proportional increase with the dose due to saturation of the transporter. So it gave us a good indication or good way of calibrating the model in healthy adults for both IV and PO administration. We did validated model further by predicting PK in adult subjects with different degrees of renal impairment. The renal elimination is the major elimination pathway for this compound. So we wanted to make sure that before we use it to predict the pediatric data, that process is really captured well with this model. And you can see that whether it was subjects with creatinine clearance close to normal, all the way down to subjects with very low creatinine clearance. So this would be in the realm of end-stage renal disease. All of those PK profiles were predicted well. So we had confidence in the adult model and used it to predict the PK in adults. For these renal transporters, we don't have the ontogenies yet. So in this case, we are looking at the predictions. First, we started with a prediction, assuming that the density or the expression levels per milliliter of tissue or per milligram of the protein are the same as in adults. And with that assumption, the predictions are not terrible, but we can clearly see that we are over-predicting a clearance a bit. Okay? And here I should point out that here we are looking really at the infants and toddlers here between about 10 months and 3 years old. So going quite far from the adults to do these predictions. And there could be some changes in the transporter activity over that long of an age. So this is where we've looked into that para-aminohippurate, right, and see, can we infer from the changes of that compound, from the changes in clearance of that compound, can we infer what might be happening with the transporters in the kidneys in general? And if we looked from this plot around 1 year old, and you can see one thing is there is a lot of variability. So it's not an exact calculation. It's really more of a guesswork as to at what range we have more of those points. So for about 1 year old, we would guess maybe about 50% of the adult. It seems to be going up around this range. So we guessed about 66% here for the 3 years old. And those assumptions would actually give a good prediction, also the clearance for that, for the amoxicillin secretion in this age group. We did have data in even younger children. And here we are really looking at the newborns or neonates, 1 through 3 years old. And on the top of it, born premature after 29 weeks of gestation. And we found in literature data for 2 different dose levels. And again, you can see we were able to scale it or get a pretty reasonable prediction of those clearances, again, by looking at this type of data for the para-aminohippurate. Again, based on this publication that I was showing also here for the slightly older children, it is a lot of guesswork, right? It's just the variability. But there was additional paper showing that kind of data for the first few weeks of gestational age. Right? So from that, we basically inferred what it might be at the 29 weeks of gestation for those newborns, and we came with an estimate of about 2% of the adult values, mainly based on this paper. And you can see it did allow us to reasonably well capture or predict the PK again, even in those very young children. So there is a potential that even as the measurements are still catching up with the actual expression levels of the different enzymes and transporters, maybe the clinical data that is already available out there for some of the other compounds which may undergo similar mechanisms, that data could be used or could be useful for estimating the ontogeny or those physiological changes as well. So this fairly long section now summarized what we are doing with the physiologies, how we parameterize the overall physiologies, how we were parameterizing some of the additional mechanisms and the principles, and also shown few validation cases in how predictive those models are, especially the models for the clearances. And the models for the distribution, right, are sort of inferred that those are working by us being able to match or predict very nicely the shapes of those different profiles through these different examples that I was showing. Okay. So to wrap up this presentation, let's take a look at some of the additional applications that we're using this complete Pbpk model framework, complete models already incorporated for pediatric applications. And a couple of these examples actually do state that they were used for regulatory applications. First of these examples, and again, it's about 6 years old now. This was a study where both Pvpk and population PK was done to with the goal of determining dosing algorithm for valganciclovir in very young children. They were looking for dosing strategy in children less than 4 months old. So they've taken this two-pronged approach, building both Pbpk model and Pppk model, just to see, are they going to be giving similar predictions? And of course, if the two different approaches meet in the middle, it gives them more confidence that it's going well. So the first paper here is showing the development of the model. The second one is then discussing the combination of those approaches and how that helped them to confirm or to come up with that combined dosing recommendation. And looking at the model itself, it wasn't really a simple or straightforward model, because it did require modeling both prodrug and the parent compound. Right? So the prodrug is valganciclovir, the active compound is the ganciclovir. The permeability limitations, especially for the parent compound, for the ganciclovir, means that all permeability limited tissue models have been used. And the transporters are involved in disposition of both compounds. Uptake transporters were needed to bring valganciclovir in the intestine for the absorption, bringing it into the tissues for also metabolism into the ganciclovir. And the parent compound ganciclovir then was dependent on the transporters to get it out of the tissues where it was formed, and also had to rely on the transporters for that active renal secretion. There was also a good rich set of data, including the animal data, that helped to determine the model structure. So it all started with the animal data that helped to determine these specifications, that we need permeability limited tissues and the transporters. And the adult human data was then used to refine the parameters. And once the adult model was calibrated, that model was then used to predict the PK in children because some pediatric data was already available. So the pediatric data that was already available was used to further verify and validate the model and give more confidence in extrapolating or doing the predictions for those very young children. This is a snapshot of few of the subjects from one of the clinical studies for which the data was available. This was a study in 1 through 16-year-old kidney transplant recipients. And this is showing a snapshot of few of those subjects ranging from 1 through 15 years. And you can see that within this age range, the predictions of the PK were very nice. All the way, from 15-year-old all the way down to 1-year-old. The model predicted very nicely, showing that by that 1 year, the transporters that were involved probably already reached adult levels. This was showing data from another study where the PK was measured in very young, in neonates. And these were all up to about 1 month old. And again, showing only snapshot of few of these subjects where for the 1 month old, the predictions were typically close to the observed data, even though I know this one does not seem like it, but there were other patients for about 1 month old that were predicted closer. So up to that point, it was okay, but less than 1 month old, there were some under predictions or more systematic under predictions of the PK, showing that maybe the transporter within the first month is still maturing and the expression levels of those transporters are lower and would be resulting in lower clearance. So a theoretical ontogeny function was developed back then, and the red points or the red lines are now showing what would be the prediction with that theoretical ontogeny function. And so to complete the story, both Pppk and Pvpk models were used to discuss the dosing recommendations for children less than 4 months old with the regulatory authorities. And they felt that the confidence in these predictions were sufficient, and it is now included in the prescribing informations in Europe, Australia, and the United States as well. The interesting point here was that different authorities actually had different views at the models, and at that time, it seemed that US FDA was a little bit more interested in the Pppk model, in the population PK model, but the Australian authority was more interested in the Pbpk model. But again, it was this two-pronged approach which gave the company more confidence in going with those recommendations. The next study is looking at the pediatric formulation development. And this one was not used in regulatory submissions, but does provide some guidance and investigations into how to scale or what to think about when doing the evaluation of pediatric formulation, okay? And how to predict that the formulation that you are predicting or designing specifically for children, how to predict the performance of that formulation itself. So what they have done here, they've done study in adults, in fasted, in fed with a typical meal in adults, but also with infant formula fed adults. And they've done a prediction first with just the default physiologies, and you can see that there were some mispredictions here due to the gastric emptying. So what they did was they actually fitted the gastric emptying for each of these scenarios, and then applied that scaled or that fitted gastric emptying and scaled it into the infants for which that formulation was targeted based on the caloric needs for that population, right? Based on the differences in caloric needs between adults and infants. So for the fasted gastric transit time or stomach emptying, they use the same value for infants, and you can see it matched very nicely. For the fed adults, they scaled those fitted gastric transit times based on the differences in the caloric needs between the adult and infant. And as you can see, doing that strategy, the scaling from infant formula fed adults, it predicted the infant PK very nicely. Adult or using the standard meal for adults didn't predict the infants nicely, but again, infants would not be eating a steak, right? So that does make sense that if the formula affects how the formulation behaves or how that pediatric meal or infant formula affects how that formulation will behave, it will give better indication of the PK. The gastric transit times, though, they also made a point. It's not that the default gastric transit times that are in the program, it's not that they are bad, right? In this case, it could have been the formulation itself that was the reason for changes in the gastric emptying. They speculated that maybe it was the excipients that are changing the thickness or the composition of the formulation. So the formulation was not emptying at the rate as we would expect for just a simple suspension or solution, but was taking a bit longer to empty from the intestine, from the stomach. Again, something that should be taken into account when designing that formulation. And the other aspect is, between these two meals, again, they speculated that the mixing of the formulation with the meal could also impact the gastric emptying and could also be a reason why the infant formula fed adult data predicted better the pediatric or the infant PK than the standard meal in the adults. This next application on the development of the pediatric formulations actually does have or did have some regulatory implications as well. This was a case study where they were developing a formulation, pediatric formulation for Strycel, for the dasatinib. In adults, it is a tablet formulation. For pediatric patients, to make it easier with swallowing, they were developing oral suspension. And they did a BE study in adults between these two formulations to evaluate how well or whether it is bioequivalent to the original formulation that was already approved in adults. And that BE study actually showed the differences. And so in this case, they've used GastroPlus. They did not necessarily have to do pediatric predictions, but they did need to do mechanistic simulations, looking at the specific mechanisms that are affecting this compound to figure out why the PK for those two formulations were different. This was, dasatinib is a BCS class 2 compound. It is a weak base, which exhibits a strong pH-dependent solubility. So the gastric emptying time or the time of residence in the stomach is going to have an impact on how much of the compound dissolves in the fasted stomach versus when it moves into the intestine. And through the mechanistic modeling, they found that it wasn't actually the formulation that was different, but it was only the gastric emptying, where the suspension formulation, of course, is going to be emptying from the stomach at a different rate than a tablet. And that was what created the difference in the BE or in the PK when they did the study with those two formulations. So in this case, even though they didn't do specifically pediatric simulation, right? Even though not in this publication, as they infer they did also simulations in pediatric subjects, just not in this specific paper. But even these simulations already were impactful on or were helping them to inform the application to approve the pediatric suspension formulation. So sometimes even just the simulations in adults and doing a mechanistic simulation helping you figure out what is happening with the compound, even without doing the pediatric, can already have an impact also on the pediatric drug development. And the last example that I wanted to point out here is, again, a little bit more looking at on the formulation side. This is actually from a group at the FDA, which of course is not to be taken as them doing recommendations. It's just the recommendation on how the Pvpk modeling could be used to help with designing the dissolution safe space, and designing how variable the dissolution rates could be for children of different ages to still have a correct performance. And it was example with oseltamivir. We've talked already about oseltamivir as one of the early applications. Here we are coming back to it with this more recent paper only 2 years ago, where again, the model includes the PK of both parent compound as well as the metabolite oseltamivir carboxylate. The model was calibrated and validated against the adult data, both IV and different PO administrations. And then the model was used to evaluate, actually it was also verified by predicting the PK for the pediatric data or the clinical studies in children that were available by this time. And then they used that model to do virtual bioequivalence explorations into the dissolution rates of the formulation and how much slower the dissolution rates could be for the formulation to still be bioequivalent. So basically they were looking at the lower range of the dissolution profile. If the basic dissolution profile is here, how far we could go here on the lower end to still have a bioequivalent performance for our formulation. And they found that, oops, apologies, I didn't mean to do that. So, and they found that while in adults, the lower or the slower formulation or the dissolution rate could be up to 10 or 12% lower than than the standard average dissolution profile to still be bioequivalent. But when it comes to adolescents, that range was narrower, going down only to about 6-7%. And when going down to the neonates, 0 to 2 months old, this was now only about 4 to 6%. So this does show that, or does provide additional food for thought, what additional aspects you need to be thinking about, and what are additional ways you can use the Pbpk modeling and the mechanistic absorption modeling to really evaluate your formulation, your products, to ensure that those children do get the proper dose of the compound and mitigating any risk on formulation and batch changes. So to sum this up, I'm hoping that by now I have convinced you that the Pbpk models are a critical tool in pediatric drug development. Dose selection, trial design, but also evaluation of pediatric formulations. Usually when you see publications, a lot of talk and a lot of focus is on pediatric dose selection. But as I've shown you here in these examples, evaluating the formulations is just as important aspect. And this has been also recognized by regulatory agencies around the world. And from the guidances that they have been issuing over the last few years, whether they are related to Pvpk modeling in general, that include comments on how to use it for pediatric designs or pediatric drug development, or the specific pediatric Pvpk guidance. It seems that the agencies are not only willing to consider or to look at the Pvpk models, but they are actually expecting that the Pvpk models will be used in determining some of these designs. Softwares like GastroPlus make this application very easy by incorporating all of that physiological information already. So you don't have to go and worry about how the liver size is changing, how the child is growing, what will be the kidney size or GFR at 5 years old versus 7 years old. All of these algorithms are already in the software. All you need to do is decide what physiology or what age or age range you want to simulate, and the program will create those physiologies based on the information that is available in literature. And as a side note, there is also an option to actually account for the growth during the course of the study if you are doing a longer-term study, or if it's on the very young children where even 2 or 3 day dosing could make an impact. We've also discussed that it's not all completed yet. Some of the processes are not well characterized yet. There are still gaps in intestinal physiologies. The ontogenies of enzymes and transporters are still being measured, and they are coming up very frequently. I showed only very small section of some of the papers that are coming up. And we do keep updating the physiologies. So it's not only that it's easy to create them, but they do keep getting updated. So the latest information is always included in the program. And we've also explored that as the information for the processes where the information is still missing, I've also shown a couple of examples showing, could we utilize the data for the clinical data that is already available, that is already out there in children, to figure out or to infer or to guess the ontogeny for the processes that are not exactly characterized yet, that are not exactly measured yet, and that those could help us to do predictions for the new compounds, for the additional compounds, by guessing how the physiology might have been changing. And that wraps up this presentation. Thank you all for your kind attention, and I believe we are still going to have a few minutes for any questions that you might have about this presentation, or if there is something else that I might have forgot to cover, or you have other questions on the pediatric Pvpk modeling.
OK， 那非常感谢大家参加本次的会议。Thank you very much for the presentation from Vera. So Vera and Hein could say hello to the audience.
Oh, hello everyone.
Thank you. Yeah, before starting the Q&A section, I will briefly summarize the content of this presentation in Chinese. 本次会议呢，我们主要是讲解了 Pvpk 模型在儿童人群的建模中的一些应用。Vera 的话呢，主要是从3个方面对 Pvpk 的应用做了讲解。首先的话呢，是对 Pvpk 的作用以及各国法规部门呢所颁布的涉及儿童 Pvpk 建模的一些指导原则。那接下来呢，对儿童的生理学的获取、规律的统计 以及目前的 GastroPlus 可以应用的模型做了非常详细的和深入的介绍，对儿童人群的组织器官的生理学、肠道生理学、代谢酶的表达、转运体的表达呢，然后这些参数以及获得这些参数之后进行的规律统计呢，做了一个非常详细的介绍。最后呢是讲解了2个具体的应用案例。第一个例子呢是采用 Pbpk 模型来指导儿童人群的剂量选择，第二个呢是采用 Pbpk 模型来优化儿童制剂的处方开发。那接下来之后呢，我们就将进行今天的这个答疑的环节。Now we start the QA section. First, we will answer the result question before the meeting, and then we will answer the timely question during the webinar. So please, Vero, could you share your screen?
Yes, you should be able to see the slides, and here is the first set of questions. Okay. So the first question was, what data is required for the Pbpk modeling, and what is the minimum sample size? I think from the presentation, you probably already guessed that there is no simple or single answer to this question. It is all going to depend on the type of compound that you are working with. The more complex mechanisms, the more of the adult data you may need in order to be able to validate the model. Make sure that you are understanding the mechanisms that the compound undergoes, and that increases your confidence in doing the pediatric predictions. For just doing the prediction in the children, you don't need the clinical data, right? The pediatric clinical data. That's what the model is for, to help you do the prediction. But once the data starts coming in, let's say you start having data in older children, it is always good to put it back into the model, verify that model, or potentially refine it before you use it to start predicting younger and younger children. Go ahead, Hank.
Yeah. 如何从这个什么样的成人和这个或者儿童的数据需要来建立这个 Pbpk 的模型？通过今天的这个presentation， 大家可能也都已经就是说有一些大体的idea。具体的实际上它是并没有一个简单的这样一个指导的这个具体的指导，它需要基于你是什么样的一个化合物。如果你的化合物比较复杂的话，它有复杂的比如说这个代谢或者吸收的途径的话，这样你就需要了解这些代谢途径，你也需要更多的成人的数据来进行验证。对于这个儿童人群，你一开始是不需要有这个临床数据的，但是在因为我们是从这个成人外推到这个 Pbpk 的模型，但是当你有更多的儿童数据就是说出来的时候，比如说经过有更多的儿童这个临床数据的时候，我们需要把它也用来作为模型的验证。比如说你有这个比较年龄大的这样一些儿童的数据，然后可以帮助我们验证这个模型，进一步优化我们的模型，这样我们就可以增加我们的这个confidence， 增加我们的自信，然后用这个模型来预测越来越小的这个儿童人群。
The second question was if adult PK data is not available, is animal developmental data, so data in adult animals and juvenile animals, helpful to do the predictions in children? So the animal developmental data would help you verify your mechanisms. Right? If you know how the specific process is changing in that animal, right? It does give you a confidence that you understand what the compound undergoes in the body, which process is the critical one, and so on. Right? But then you still need to be careful about translating the animal data into children. You need to account for the animal to human prediction, as well as the ontogeny in human is not going to be the same as ontogeny in the animal. So again, it could help you to increase your confidence that you understand the processes and the mechanisms, but the translation into the children, make sure you are using the correct animal to human translation.
动物学的数据，它可以帮助我们，就是有一定的帮助。比如说，尤其是帮助我们来确定一些比较重要的这个机理。因为比如说，如果我们知道了这个动物的这个发育的这个数据，然后帮我们验证了某一些比较重要的这些机理，然后在这个不同的这个动物，比如说动物这个年龄中间，我们可以看到，帮我们验证这个机理的重要性。但是我们要注意的话，就是建立，如果我们的儿童人群的话，我们还是要知道这个儿童这个就是这个人群里面对于这个机理的它的这个发育的数据。所以说我们在用动物的数据做外推到这个儿童数据的话，它动物的数据最主要是帮助我们确定这个机理哪一些这个机理是重要的，确定这个重要的机理。但是就是说我们要比较小心，然后如何从这个动物的这个发展数据到儿童发展数据，儿童的这个我还是要有自己的这个人类的这个发展的数据来进行。但是动物的数据和动物的这个 PK 的数据可以帮助我们验证比较重要的这个代谢的机理或者吸收的机理。
Okay. The third question was how to calculate the dosage for children and what data and information is needed to build and validate the model. There was a lot on this topic, especially the second part of the question, how to build and validate the model in the presentation itself. Right? Typically, you would have some animal data and human data that help you to verify that you understand the mechanisms. Right? You want to make sure that you are able to model different scenarios in adults. Right? Let's say different dose levels, single doses versus multiple doses. If there is data available for different administration routes, right? All of that gives you the more complete picture about the compound, what is happening to it in the body. And if you are able to model all of that with a single model, that gives you confidence that you understand your compound and that you are really capturing the important mechanisms. And you can start doing prediction for children. And if you are interested in calculating the dosage for children, one additional assumption or estimate you need to make is what is the exposure that you need to have in children? Is the plasma concentration, does it need to be in the similar range as in adults? Or is there a difference in the disease where the concentrations in children need to be different? Right? So that comes down to understanding the disease. And once you have the model and you have the target exposure in children, then you will use the model to figure out what the dose will get you the exposure in children that you are looking for, that you need.
对于尤其是这个问题的第二部分，需要什么样的数据和信息来建立和验证的模型，这也是今天报告这个主要讲的。首先你需要利用比如说是动物或者是成人的数据来建立的模型，然后是建立的模型以后，然后我们需要用成人的这个数据，各种的数据，各种的比如说不同的这个给药的剂量，或者是单次给药或者多次给药，就是我们希望用一个模型，一个模型能够这个模拟或者是验证这所有不同的给药这个情况。这样我们就可以全面的理解我们的这个药物。如果我们全面理解了这个药物，然后可以用一个模型来验证这个所有的这些成人的数据的话，这样我们就会有信心来这个预测这个儿童的这个建立儿童的这个 Pbpk 模型。对于问题的第一部分需要什么样的剂量，这首先是要基于我们是你希望达到什么样的这个生理学的这个药物的这个 PK 的这个 data， 或者是这个药物的浓度，你希望的这个目标的药物浓度是多少？你是希望它和成人的是一致的，或者是说基于你对一些疾病的了解，你知道比如说这个儿童的这个给药量儿童的这个目标的这个药物浓度和成人的是不同的，如果你对这个疾病有足够的了解的话，那样我们根据就是我们需要达到的这个目标的这个药物浓度，那我们就可以利用 Pbpk 模型来就是预测或者计算出我们需要这个儿童的给药剂量。
The fourth question is how to assess or validate the accuracy of the dose prediction in the pediatric populations. This comes down to once you start collecting some of the data in the pediatric populations, you compare the prediction with the observed data, right, with the plasma profiles. The one thing I would like to point out here is be careful about comparing simply Cmax and AUC. In children, and especially as you start going into the younger groups, you may not be able to take as many samples as in adults. You will have fewer samples, and you may not be able to completely capture the Cmax and AUC. For example, this is what a typical CP time profile would look like. But you are doing a study in a young child, so you can only take 4 or 5 samples. Right? And you take a sample here, maybe here, the next one comes here, and then you get samples here. Okay. If you overlay the simulation with the individual points, it is a very good match. Okay. But if you tried to compare only Cmax and AUC, okay, it would look like the model missed the Cmax prediction. But that is not a problem of the model. It's simply that the Cmax wasn't measured. Right? So just be careful and consider what you actually measured and how much sampling you were able to do and which parameters make the most sense to compare to the observed data.
Yeah. 当你开始采集儿童的数据的时候，你可以就是用你采集的这些药物的浓度和这个模型预测来比较。但是当我们比较的时候，我们要特别注意，不能够简单的用这个我们成人经常用的验证 Pbpk 模型的时候用的 Cmax 或者是 AUC 来进行比较，因为对于儿童人群，我们不能够像成人一样有就是说足够多的这个采样量。这里 Vera 给了一个例子， 对于这个成人，我们可能有多个这个采样，但是对于儿童人群来说，对于一个这个儿童的话，我们可能只能采4到5个这个采样。如果我们用这个红线来作为是我们模型的预测的话，然后这些蓝色的点是作为我们这个采样这个实测值，然后我们可以看到，实际上整个的这个曲线和这个数据量之间是有很好的拟合。但是如果我们只比较这个 Cmax 或者是 Tmax 的话，我们就发现，你可能就会认为，或者是 AUC 的话，你就会认为你这个模型的预测并不好。但所以说我们要特别的小心，在我们就是验证这个 Pbpk 模型预测和实测值之间，然后我们要首先要考虑到这个儿童人群，我们并没有足够的采样量，所以说我们要这个首先确定要怎么样，就哪一些这个判断标准来判断它是一个这个来进行验证。
Well, this question actually directly follows the previous discussion. How to design the sparse sampling to support the generating model inputs prior to Pvpk modeling. You should actually be doing it the other way around. Do the Pbpk modeling first before you do the study, and that also gives you information about the sampling, in which region you should be sampling more. And then when you get the data, then plug it back into the model to verify or further refine the model. But you really should be starting the Pbpk model should be the very first thing you do before you design a study.
实际上对于这个我们这个顺序应该这个问题问的是在建模之前如何获得这个儿童数据。实际上我们这个顺序应该改变，在获取采样，在获取这个任何儿童的采样之前，要先做 Pbpk 的这个模型，然后根据 Pbpk 模型的预测，然后这样我们来设计我们的儿童的采样。比如说我们就可以确定在哪一些这个时间段，或者在哪一些这个时间内，我们要比如说采样比较那些就是比较重要的部分，然后再利用我们采样以后得到的数据来进一步就是来验证和优化我们的模型。所以说这个我们要这个顺序要有改变，就是说对于在儿童数据采样之前，我们要先做的是 Pbpk 模型，然后来通过 Pbpk 的模型来帮助我们设计这个儿童的这个采样，就是采样点，然后再利用采样的数据来这个优化我们的 Pbpk 模型。
Question number 6 was how to evaluate and improve the accuracy of the model prediction if there is not much data. And, that is one of the reasons why we are doing predictions, because it is not easy to generate the data. You will simply use the data that is available. That is one of the very common uses of Pbpk models, is to build a model so that you can do a smaller clinical trial in children. So you don't have to enroll as many subjects. You would only enroll few to verify the model, and then supply or do virtual population simulation using the Pvpk model to account for wider variability. But if you don't have any data, of course, you cannot evaluate or compare the model. You simply use the data that is available.
Yeah. 在这个儿童数据不多的情况下，所以说我们更需要运用这个 Pbpk 的模型，然后通过 Pbpk 的模型，然后我们可以就是用就是说 婴幼，就是说我们可以用比较少的这个儿童来做这个得到这个 PK data 来进行这个模型的验证，然后通过验证的 Pbpk 模型，我们可以来预测在各种不同的其他更大的这个 viability 的情况下，在这个来进行它这个模拟的这个 virtual BE 的情况下virtual simulation。当然就是说，如果你在这个正常情况下，我们只能用我们这个不多的这个数据来进行这个验证，然后我们可以用的就是这个 Pbpk 模型来帮我们，就是说我们用这个设计实验的时候可以用比较这个少的这个儿童进入这个研究，然后用得到的比较少的这个数据帮我们验证这个模型，然后通过这个模型，然后再可以预测到在在更大的这个人群之内的这个有更大的这个 viability 情况下的这个PK 的 data。
How do Pvpk models predict the effect of food on the PK in pediatric population? This is part of the model where we have a bit more uncertainty because the intestinal physiology in children is less well characterized. There are still gaps in knowledge about what the intestinal physiology in children looks like. But there also the other aspect is you are not designing studies fasted versus fed, right? You are doing these studies in patient populations, and oftentimes you may not have an option to actually do a fasted study. So there isn't much data available to evaluate very clearly what that would look like. However, there was, let me open very quickly the slides that you just watched, because one of those examples presented at the very end actually touched upon this topic. Okay? And that was this example for the development of pediatric formulation, where they actually evaluated the food effect in adults first, and then they used the calibrated adult model and scaled the relevant physiological parameter. In this case, it was the stomach transit time from adults to children to predict the food effect and to predict both fasted and fed condition also in children. So this study actually did give an idea about how to look also at that type of effect. They specifically looked at the gastric emptying, but similar strategy could be used for other types of compounds.
Yeah, 对于这个食物的效应来说，是我们有在这个部分还是有一定的不确定性，因为通过这个报告我们也知道，对于儿童人群肠道的这个发育情况还是有一些这个不是很清楚的这个机理。但是在这次报告中，我们也可以看到这一页这个显示的这个例子，它也在这个例子里面，它这个首先通过这个成人的人群来计算出，就是说对于这个胃的这个传送这个时间。然后通过得到这个在这个食物情况下，这个和这各种不同的情况下，这个胃的传送时间，然后这个推算到这个在儿童人群中，然后我们可以看到对儿童人群的这个预测。在这里我们看到，它最主要只是改变了一个参数，就是这个胃的这个传送时间。所以说就是在这个例子里面只是改变了这个参数，但是就是说最主要的这一个问题就是属于在食物情况下，就是因为我们对儿童的这个肠道的这个一些这个参数 并不是特别的清楚，所以说对于预测这个在实物情况下呢，还是有一定的不确定性。第二问。
So let me go back to the questions. What is the current status of application of Pvpk modeling for monoclonal antibodies, antibody-drug conjugates in children? This is an interesting question. There were actually 2 papers in the most recent AAPS journal on this topic. One of those was looking at the predictions of Humira, or doing dosing for Humira for children in regulatory submission. That model did not use Pvpk though. That one was still based on population PK, but it shows the overall goal of the FDA and using this model-informed drug development strategy to help in other areas, including the monoclonal antibody. The second paper was actually a review of cases where the monoclonal antibody PK was predicted for children. And they were able to find in literature only about 6 or 7 cases where Pbpk models, different types of Pvpk models, were used to predict the PK in children based on the adult PK of monoclonal antibody. And those predictions actually came out quite reasonable. They were usually within the range of predicted variability or within 2-fold of the observed data. So it's not perfect, but very reasonable predictions were obtained with Pvpk models. We have not completed those simulations yet with GastroPlus, so I cannot tell you the accuracy of the GastroPlus models. But in that paper, they highlighted, of course, a lot of gaps that are still in terms of knowing the ontogeny of parameters. But even with those gaps, the models are already able to do a pretty good job on predicting the PK for monoclonal antibodies as well.
Yeah. 最近对于这个问题，大分子这个药物的话，最近在 AAPS 上面发表了2篇文章，一篇文章是关于这个 Humira 这个药物，然后它是提交给 FDA 的，在这个例子中间，它没有用到这个 Pbpk 的这个建模，用了其他的这个模式。另外一篇文章，它是一篇这个 review 的文章，然后它这个在发现了在现有的已经发表的文章中有6到7个这个应用的案例，就是从这个用 Pbpk 模型，各种不同的这种 Pbpk 模型，从这个成人推算这个儿童的药物，然后整体的这个预测的结果还是不错的，然后都是在这个viability 的这个范围之内的，对，然后如果大家感兴趣的话，可以去看一下这2篇文件。
Okay, so going to the next page. What is the biggest difficulty in predicting the pediatric population compared to adults? That really depends on what compounds or what type of compound you are dealing with. You might recall some of the information presented in the webinar. One is there are still uncertainties about some of the physiological parameters in the intestine. So those might be adding more uncertainty on the predictions. Of course, the ontogeny of transporters. Right? And again, I was describing there some of the papers that were coming up, and there is a lot of literature coming up recently on expression levels and ontogenies of transporters and enzymes, but they are still not completely characterized. So for some compounds, it's actually quite simple and quite easy to do because the physiological information is available. For other compounds, there might be more difficulties, but it really comes down to the gaps in the physiology that we are still trying to fill in the knowledge that is still missing.
对于这个问题来说，最大的难点还是基于你的这个化合物，你的药物。对于有一些药物可能就是比较容易预测，因为对于它这个，比如说它的一些这些生理学的参数，都已经比较清楚。但是对于通过今天的报告，我们也可以看出，对于有一些例子来说，比如说对于有一些药物，它可能涉及到对于一些我们现在还不清楚的一些这个肠道的这个发育的生理学参数，或者说对于有一些转运体的这个发育的生理学参数现在还不清楚的话，然后这样就是建模了起来就会比较难。现在最主要的这个还是依据于就是说我们现在对于这个还有一些 gap， 就是说还有一些这个对于这个一些生理学参数可能还并不是特别清楚。尤其是对于你的这个药物，如果涉及到这些不是我们现在还没有这个研究清楚的这一些生理学参数的话，可能就会建模的时候就会要比较困难。这时候我们就需要增加一些这个通过这个验证，可能我们要进行一些这个模型的优化。
What are the main differences between the pediatric and adult models? When can the models be extrapolated to children? I think that is actually very similar to question 11. What types of drugs are suitable for adult to child extrapolation? So the main difference is availability of the data. And you have much richer data sets in adults. Oftentimes you have data in healthy subjects, single dose levels, multiple dose levels. Then you have data in patients, potentially also DDI data. So a lot of information as you are building the model, as the drug goes through the development process, and you are running new and new clinical studies in adults, all of that data comes back to the model to help to improve it in adults. So by the time you get to pediatric predictions, you understand the mechanisms. If you were starting directly with the pediatrics, right, you will not have clean data in healthy children. Right? It's going to be only patients. You cannot do a clinical study in children to just dose the drug and do the PK. It may not be as well controlled as you can do in the adults. So it is a lot more messy data and would be more difficult to actually develop the model based on pediatric data alone. It can be done, but it's just easier to do it from adult data from more well-controlled studies. And when can the models be extrapolated to children? You could do it for any types of compounds. The confidence in the prediction may be lower for, let's say, compounds which are substrates for transporters, which have, or formulations that may have complex mechanisms happening in the intestine and so on. You may have lower confidence in the prediction or may need to account for larger error of that prediction. But you can still do the prediction to at least give you some idea about what PK to expect in children.
对于这个儿童建模和成人建模主要的差异就是在于这个他们 data， 这是他们的数据，这个可以用来这个建模的数据。对于成人来说的话，我们经常这个数据是比较丰富的，我们可以有单剂量、多剂量，可以在健康人群，可以在疾病人群，甚至还有这个药物相互作用的这样一些这个数据来帮助我们验证模型。所以说，在我们建立成人的模型的时候，我们还是有比较多的数据的话，对于儿童来说的话，正常情况下可能我们不会有一些比较这个完整的这个数据来自于健康这个儿童，基本上都来自于这个疾病人群，并且我们前面也看到对于这个儿童数据的采样， 就是也并不像这个成人的这么丰富。如果我们直接建模就用这个儿童数据的话，我也是可以的，但是就是说我们的数据就不像这个成人数据那么的就是说丰富，然后并且是儿童的数据经常来说是比较这个 messy 的，是比较这个杂乱的。然后什么情况下我们可以外推到这个通过成人外推到儿童？任何情况下，任何的这个化合物都是可以的。它最主要是要基于你这个药物的机理，有可能你会有比较这个并不是有就足够的这个，只是对于它这个外推的这个模型的预测，可能是有这个 low confidence， 并不是这个最可信的。比如说这时候可能比如说如果有一些转运体。然后，因为我们现在转运体的这个发育的参数可能并不是特别清楚，有一些转运体的这个设计的一些药物，或者说对于你的这个剂型特别复杂，然后这个剂型牵制到在这个肠道内有比较这个复杂的这个溶解啊，或者这个吸收的过程，可能也会导致我们对于这个外推的这个预测会有一些 low confidence。但是正常来说的话是所有的如果你建立了用成人的数据建立的模型，都可以进行儿童的外推。然后只是对于这个外推的这个模型的预测的这个自信的这个程度，基于这个药物的机理可能会有不同。Sarah.
What factors need to be considered when extrapolating from clinical PK data without healthy subjects? Right? Can this be only extrapolated to healthy pediatric subjects? You will never be extrapolating to healthy children because you will not be dosing the drug in healthy children. It's always going to go into patients. And that is the beauty of the Pvpk model, because it can account for differences of in physiology, not only between adult and child, but also between healthy child and a sick child. It all comes down to knowing what the physiological difference is. For example, if your compound is excreted in urine, and you know how the GFR is changing over the ages, how it's changing between adults and children, of course you can scale it into children. But now if that child or the children that you will be treating are also going to have renal impairment and will have lower renal secretion, again, something you can account for in the model and further modify it to scale into patients and not healthy children. So the factors that need to be considered are the physiological differences.
在正常情况下的话，我们进行儿童的外推，实际上我们预测的就是属于疾病人群，所以说在这里面就是我们可以考虑到这个Pbpk 模型的这个优势。它不仅考虑到这个生理学参数的变化，从成人到儿童，它也要考虑到就是说从这个疾病人群中这些不同。然后比如说在这个生理学这个参数的不同，比如说如果这个药物最主要是通过这个肾脏通过这个来排泄的话，这时候不仅通过我们有这个参数这个 GFR。这个在随着年龄的变化，然后我们可以考虑到这个疾病对它的影响。如果我们知道疾病对它影响的话，我们就把它这个生理学参数的变化加入这个 Pbpk 的模型的话，这样我们就可能预测出在这个疾病人群中，这个疾病儿童人群中的这个预测。
Okay. I think this is the last slide. Which pediatric clinical studies can be waived? I think you probably have more success with waiving the studies in older children because they are closer to the adults, less extrapolation being done. But there have been cases of waiving or being able to do a smaller trial for also younger age groups looking at other doses. I did share a couple of examples which did not completely waive the study. For example, this was one which on the valganciclovir on the val side, it did not completely waive the study in those children less than 4 months old, but it allowed the company to do much smaller trial. So the trial or the study could be completed in much shorter time because they didn't have to enroll too many children. They only enrolled few to verify the model, and it helped to inform the dosing or the labeling of the compound. So there is no simple answer to that question. It all depends on what kind of compound you are dealing with and what kind of model you are able to build based on the information about your compound and the physiology.
比较容易，这个可能就是说waive 掉的一些 study， 可能对于一些比较年龄大一点的儿童，对于比较这个青少年来说的话，比如说大一点的这个年纪的儿童来说，因为他们的生理学参数可能跟成人的变化并不是特别大，有比较小的这些变化的话，比较容易这个可以这个 waive 掉。但是另外一种应用的话，就是说你可以通过 Pbpk 的模型，然后这个你可以争取只用比较少的这个一些这个临床实验，就是说我们只用少量的儿童参加了这个临床实验，然后就是来帮我们验证这个模型。在这一页这个报告中Vera 就给了大家一个例子，然后就是说对于这个比较小于这个4个月的儿童来说的话，然后我们只需要就是说少量的儿童得到这个临床数据来帮我们验证这个模型，然后我们然后这样我们的这个实验可以比较短的时间内这个完成，并不需要就是说就是说需要大量的儿童来进行这个临床实验，所以说它最主要还是要基于你的这个化合物，你的这个药物，它这个机理，还有就是说哪一些生理学参数可能会比较重要，然后可能就是有不同的情况。
Next question. In which stages of development of children products are the Pbpk models more widely used? This is actually something that definitely at least the EMA and FDA guidances do have the requirement of this pediatric investigation plan, which needs to be put in place quite early. I believe it's around phase 2, but I'm not sure about that one. But those are the stages where they would already be looking at the Pvpk models. So very early on, when they have to plan for the pediatric studies, the Pvpk models for children are being used and developed. The Pvpk models themselves start getting developed on day one. Right? That's how you should be using the Pvpk. Building it as you are generating the data, keep improving the model so that when you get to that stage that you need to design the trial or communicate it with the regulatory agency, you have that model ready to go into doing pediatric predictions.
Yeah， 在这里面就可以看到，对于 FDA 和EMA 的这个他们的这个指导意见中，也可以看到他们在比较早的这个阶段就要求你要提供儿童这个实验的这个计划，也就是说可能是在这个 phase 2的情况下，你可能就需要提供这些这个结果，所以说对于这个 Pbpk 模型在儿童中的这个建模的话，我们还是希望就是说在越早越好。 然后这样的话，你可以不断的这个优化和验证这个模型。然后当你就是和这个 FDA 啊，或者是和其他的这个监管机构进行这个讨论的时候，然后这样的话，如果需要提供这个儿童的这个实验的计划的时候，你就可以有这个 Pbpk 模型的预测。
Are there any pediatric Pvpk models successfully used in the submissions? Have the results been included in the drug label? Again, there were a couple of examples, and this was one, right, which was, and this actually goes also to the last question, which was used for dosing algorithm for children less than 4 months old. And the information is in the labels, in the drug prescribing information. The interesting part for this one was that for this specific case, it was actually the Australian agency that was a lot more interested in Pbpk model. But there are other cases also where the Pvpk models have been used with both EMA and FDA as well. Another one was, I think, this case where I'm not sure if this had anything to do with the label, but again, evaluating the formulation did help them to make the regulatory application and the regulatory submission. And again, these are only a couple of examples. Pediatrics are now the Pvpk models are now quite commonly used for either design of the trials or the pediatric predictions.
对于这个问题来说的话，在今天的报告中也给了2个例子。在第一个例子里面，我们可以看到，对于这个比较小的这个婴儿的话，在这给药中我们建立了 Pbpk 模型，然后它也在这个药物的这个 label 中也体现了出来。另外一个就是这个例子，然后对于这个模型的话，然后它是澳大利亚的这个监管机构，然后是对这个 Pbpk 模型这个比较感兴趣。另外还有就是说其他的一些药物，我们可以看到也是在这个 EMA 和这个 FDA 中间，就是也是有一些这个应用的。另外一个例子。然后是它是在这个和这个监管机构这个提交文件中有的，然后但是就是我们并不知道是它最后有没有进入这个label 中。现在我们也可以看到，对于这个儿童人群中的 Pbpk 模型的应用也越来越多了。
Okay, and the last question was about the Pvpk models for infants, neonates less than 2 years old. Okay? Again, it can be done with Pvpk models, and this is one of the examples that I keep coming back to. Okay? This was the model where they were specifically looking for the dosing algorithms for children less than 4 months old. And the models did help to inform those dosing algorithms and the decisions. And this wasn't even one of the simpler compounds. Right? This was a complex model which included a prodrug and the parent compound. It had transporter involvement, permeability limited tissues. So not a simple compound by any means, and yet it was used for children less than 2 years old. And there are other examples. I'm sure we could find more examples in the regulatory submissions.
Yeah. 对于小于2岁的这个儿童来说，这个 Pbpk 的模型是这个可以建立的。然后在这里我们就是可以看到，同样是这个例子的话，然后是最主要是需要来预测对于这个小于4个月的婴儿这个疾病人群中的这个给药。在这里我们看到，实际上它并不是一个简单的这个化合物，实际上这个化合物的代谢是非常的复杂的。它首先它是有这个 prodrug， 然后并且还有这个parent 的 compound， 并且它也是用到了这个 permeability limited 的，是有这个 并且还有转运体，也这个就是也这个机理也用到了，实际上它是一个特别的复杂的一个机理，但是对于这个就是建立的这个 Pbpk 模型也是被这个监管机构，它的预测结果也是被监管机构所接受的。
I think that concludes the questions that we had. Let me find the slides.
Yeah.
Yes, that was the last.
Yeah, I think there are the question, the other two questions from the webinar. One is like except using the adult model for build the adult model first, then extrapolate to pediatric. Could we consider to use like the pediatric model from other country or other population, and then use the own countries the pediatric data and to build the model like or to the optimization or validation. So I think the question is, could we use like the data from other countries pediatric data and extrapolate it to another country's pediatric?
Yeah. The strategy would be similar to extrapolating from adults, right? The only difference is that instead of looking at the ontogeny of the process, how those processes are changing between children and adults, you would be looking at the differences between different populations. For example, if is the 3A4, right, different in European children versus Chinese children, right? Those are the differences that you would account for. But in those terms, the Pvpk model could be used in those scenarios as well.
Yeah.
I don't have a specific example where it was used, but in principle, there shouldn't be any difference than going from adults to children.
Yeah. 对于是否可以考虑从其他外国人群的儿童模型作为基础，经过本国儿童人群的数据进行优化和验证，来进行种族间的外推和建立。Vero 的这个回答是，实际上它是和成人外推到儿童是类似的。我们首先要知道它生理学的不同，然后我们要知道就是外国人群和本国人群之间生理学的不同。比如说一个例子，比如说对于 3A4 来说的话，对于这个白种人和这个中国人群中间，比如说是不是儿童这个人群中间是什么样的差异的话，如果我们知道这些生理学的参数，那同样的是和建立这个成人外推到儿童是一样的，然后通过其他国家的儿童模型，然后外推到本国的儿童人群，也是主要是基于就是说生理学参数，这个我们是不是知道有哪些不同？另外一个问题就是does GastroPlus have the Chinese pediatric population? And if we do not have it, could we use Caucasian pediatric population to build the model and for Chinese pediatric population?
GastroPlus actually does have Chinese pediatric population. It does include algorithms, and this is something that we do with some of the populations. Some of the information might be borrowed from the Caucasian if we didn't know what the information is, but you do have an option to build the Japanese physiologies and also do the pediatric. And I know there was a lot of information on how the body weights and heights in Chinese children compared to the Caucasian children. The tissue volumes are also specific to the Chinese children. I think this is something that Haiying, you probably have better overview of all the information that went in. For some parameters, we are borrowing the information from Caucasian children, but a lot of the information is actually specific to Chinese children.
Yeah, 这个问题是在这个 GastroPlus 中有中国儿童人群吗？在这里面的回答是我们是有中国的儿童人群， 然后在我们建立这个中国的这个儿童人群中间，我们首先就是考虑，就是说采用了这个很多中国儿童人群的数据。对于有一些没有的数据，我们可能是借用了这个白人这一些的这个数据。在这个 GastroPlus 你可以看到，你建立 Pbpk 的模型的话，你可以建立这个 Caucasian 白人或者是日本或者和这个中国人群，你可以看到就是在这里面我们考虑到了很多的这个这些通过不同的人群它本身的参数，然后另外你可以看到就有一些参数我们可能是通过这个去借用，比如说其他的种族，或者比如说在这里面我们可以看到，对于有一些这个酶的这个数据的，我们可以看到有一些我们是通过这个，因为对于这个本身的数据如果没有的话，我们可能是从这个白人的这个数据中里面借用的。对，但是就是说我们建立这个中国的儿童人群的时候，我们是尽量的，就是说如果已有的数据有的话，关于中国儿童人群的生理学参数，我们是把它都考虑进去的。
Okay, this is all the question for this webinar. So thank you, Vera, and thank you, Haiying, for this webinar. This is all for this meeting. Thank you again. 那各位参加的学员，我们今天的会议也就到此结束，也非常感谢大家的参与。OK, thank you again, and Vera and Haiying, good night.
All right, thank you everyone. Bye.